All right, welcome class. Feel free to let me know if you guys have any questions or put it in the chat. I just wanted to show you, hopefully you can see my Canvas screen up here, but you do have an upcoming quiz on chapters 1 and 2, just on these chapters that we've been going over. It'll be available tomorrow night at midnight, and it's open until Sunday. And you guys have 25 minutes to do it. It's based on the lecture chapters, and for quiz, hold on a second. For quizzes, I'm okay if you guys use your notes for quizzes, because I want you to get familiar with the material. For tests, I would strongly encourage you to not use notes, and I just do the honor system, and I expect you guys not to use notes. But for your quizzes, I'm okay with you guys using notes. But that quiz, just keep in mind, that'll be due on Sunday, but you have all weekend to do it where it'll be open for you. And then the Labster, you have another lab due on Sunday, a cell membrane and transport lab. Again, hopefully your computers are working. Remember to use Google Chrome. And I extended the due date for lab 1 until Sunday as well. And if anyone needs more time with lab 1, let me know. The quiz is just, it's 10 points, but some of the questions are like a matching. So it maybe is like 4 or 5 questions, but it's 10 points. All right. Any other questions with that before we get started on, this is the second half of chapter 2. It's more chemistry, but I promise this is the last of the chemistry. The second half of this chapter is really on biochemistry, talking about some of the molecules that are in our body. So this will be important. You'll also notice the background. I always try to use different backgrounds on my Zoom of places I've been. So if anyone can guess, the other background was from the Pacific Northwest in Olympic National Park. But if any of you can guess where the background is, it's one of my favorite places in the whole world. Put it in the chat. We'll see if you can guess it. But let's talk about some biochemistry. And again, it's important to have a good basis, especially in biochemistry, because we'll be learning about a lot of the chemical composition of molecules that are in our bodies and what our bodies need for life. All chemicals can be inorganic or organic. Inorganic compounds are your waters, your salts, many acids and bases. They will not contain carbon, so that's why we call them inorganic. Organic compounds are all of our carbohydrates, fats, proteins, and nucleic acids, and these will contain carbon. They're usually larger and they're covalently bonded. So that's the difference between organic and inorganic. Both are essential for life. Not Monterey, Santa Cruz. It's not in America. Good guess though. Water is the most abundant inorganic compound. So again, it's inorganic, it does not contain carbon. It has important properties that you can read through here. It has a high heat capacity, a high heat evaporation. What that means is it has a high boiling point. So water can get hot, really hot before it boils. It's very good at dissolving substances because it is a polar molecule. It's very reactive, and a cushioning availability just to help protect things that are in it. So this explains what the high heat capacity means. It has the ability to absorb and release heat with little temperature change. A high heat of vaporization. This will be useful for cooling mechanisms. The ability to dissolve things, specifically ionic substances that have a charge to them because it is polar in itself. So it has a negative and a positive region to it. It's the body's major transport medium. And again, this is just showing how water can kind of surround itself around the sodium and chloride ions and separate them. And that's how it will dissolve a salt crystal. It actually takes apart the sodium and chloride atoms, surrounds them, and that's how it will dissolve salt. It's very reactive, so it's necessary for hydrolysis and dehydration synthesis reactions that we'll talk about. And cushioning, it just protects certain organs from physical trauma. So there's a layer of cerebrospinal fluid that cushions your brain. So that's what cushioning means. You have water or substances that are mostly made up of water to provide protection for different organs. Salts are ionic compounds. Again, they're ionic compounds, meaning one has a positive and a negative charge to it, like sodium chloride. They will dissociate into separate ions in water. All ions are called electrolytes because they can conduct an electrical current in a solution. So you might see liquids with electrolytes added to them. Ions will play a specialized role in body functions. So your sodium, your potassium, your calcium, and iron are four of probably most of your important ions that your body needs. We need an ionic balance, so a balance between those ions to maintain homeostasis. And these are some common salts in the body. And the big one is sodium chloride. Acids and bases are also both electrolytes. They will ionize and dissociate in water. That just means acids and bases all dissociate or break apart into separate components in water. We call acids, they're proton donors, and that just means they release hydrogen ions. An example is, I'm going blank. Hydrochloric acid, excuse me, it's like the strongest acid. It will dissociate into hydrogen and chloride ions. So any sort of molecule that can release or give up hydrogen ions is called a proton donor. And these are important acids in the body. Hydrochloric acid is important in your stomach for dissolving things. Acetic acid and carbonic acid. So they are proton donors, they give up their hydrogen ions. Bases are what we call proton acceptors, and that means they pick up hydrogen ions in a solution. So when a base dissolves in a solution, it will release what we call a hydroxyl ion, which can go on and accept a proton if needed. And these are important bases in your body: bicarbonate ion, not necessarily in the body, and also ammonia. The pH scale gives the relative concentration of acids and bases. It's a measurement of the concentration of hydrogen ions in a solution. So the more hydrogen ions you have in a solution, the more acidic it will be. pH is what we call a negative logarithm of hydrogen ions in moles per liter. That ranges from 0 to 14. All you need to know is a pH. Well, you should just know the general pH scale, but in terms of how we get the pH numbers, we get them logarithmically so that each pH unit represents a 10-fold difference. So a pH 5 solution is 10 times more acidic than a pH 6 solution. So this scale of 0 to 14, each digit represents a 10-fold difference in between the digits above and below them. So acidic solutions have a very high hydrogen concentration or proton concentration, but a low pH. So acidic pH ranges are anything from 0 to 7 or 6.99. Neutral solutions have equal numbers of hydrogen and hydroxyl ions, and all neutral solutions are pH of 7. Pure water is always pH neutral. Now, keep in mind that's extremely pure water. So if there's something added to it, it could be a little basic or acidic depending on what might be added to it. So here's the pH of pure water. A pH of 7 has this hydrogen ion concentration. We say it's 10 to the negative 7 moles. You don't really need to know that. You should just know that pH neutral is 7, acidic pH is 0 to 6.99, and anything greater than 7 is what we call alkaline or basic. They will have a low hydrogen ion or proton concentration, but an extremely high pH, and it'll range from anything greater than 7 to 14.
I have a question.
Question, Mia? Yeah.
Or it's not really a question, it's just like, I thought alkaline water was like better than like pure water or regular water.
It might be better because I know they add some things to water, probably like some sort of electrolytes. So alkaline water would be slightly basic. So they add extra electrolytes to it. I'm not exactly sure what they add to it. But yeah, different electrolytes that are added, different ions can be helpful to the body. I don't know, I'm not a water expert, but I mean, that's okay. It would just be a little more basic because it would have extra electrolytes added to it.
Cool, thank you.
And you see that, like electrolytes added to smart water or life water. Yeah. You add anything to a label and people will buy it too. Have you noticed that? You just got to be creative enough for an idea and then you can make tons of money. But yeah, yes, that's true, Mia. Here's the pH scale. Again, you don't necessarily need to understand the concentration in moles per liter. I just want you to know that 0 to 6.99 is acidic, 7 is neutral, and then anything over 7 would be considered basic. Whoops, I was going backwards here. Basic is at the top, excuse me, and very acidic is at the bottom. So you'll have hydrochloric acid is extremely acidic, lemon juice, wine, black coffee, milk is slightly acidic. Blood is just over 7, it's 7.4, so which is slightly basic. Egg white, bleach, ammonia, oven cleaner, and then sodium hydroxide. So just understanding the relative pH scale is really important in biochemistry and in our bodies, because if our blood is slightly too basic or acidic, bad things will happen. Neutralization reaction, when acids and bases are mixed together, displacement reaction occurs, and this will form water and a salt. So what a neutralization reaction means is if you take a really strong base, which is sodium hydroxide, and if you mix it with a really strong acid, hydrochloric acid, they will neutralize or cancel each other out. So kind of like opposites attract. You have a strong base and a strong acid. Displacement will occur, meaning different things will interact and they'll switch spots, and you'll get a completely neutral reaction. A buffer resists abrupt and large swings in pH. So buffers are important for kind of maintaining any sort of abrupt or large swing in pH. And acidity involves only free hydrogen ions in solution. So buffers will release hydrogen ions if the pH rises to try to lower it, and they'll combine. They can bind up excess hydrogen ions if the pH falls. So they can convert strong acids or bases into weak ones. I don't know why there's extra letters here. Sorry, guys. What is that? Really sorry about that slide there. There's like an extra letter. Unless it's just showing the buffer, I think there should just be an arrow there. Sorry. But a buffer is just important in the body to resist any large change. And this is important when we get to the respiratory system. When you guys take in carbon dioxide, the release and uptake of carbon dioxide controls your pH levels of your blood. And we'll talk more about this equation when we get there. A homeostatic imbalance. So this is a little bit about the pH levels of the blood. Your enzymes in your body work within a very narrow pH range to keep chemical reactions occurring at the right amount, the right rate, and to keep everything normal in the blood specifically. If you have an arterial pH of 7, which is just under the normal 7.4 of your blood, so this is talking about arterial blood, if it's slightly less than normal pH of 7.4, if it's at 7 during some sort of cardiopulmonary resuscitation, so someone's going into cardiac arrest and they're trying to resuscitate them, this will predict a poor outcome if pH levels fall to 7. And if a patient presents with an arterial pH of less than 6.85, they will rarely survive. So again, when we're talking about arterial pH, we're talking about blood pH in the arteries. Normally it should always be held at 7.4. And if it gets less than 6.85, patients will rarely survive. So you have the body's way of trying to maintain that. It can be due to increased carbon dioxide. Yeah, so we'll talk about how when carbon dioxide gets into the blood, it will turn into, or it'll dissociate into carbonic acid. So your body will go into different respiratory strategies, and your kidneys will kick into gear to try to increase or decrease those levels of carbon dioxide. It'll get interesting because we'll start to talk about deep, shallow breathing to try to keep carbon dioxide in the body, or excess breathing to try to get carbon dioxide out of the body to control those levels of pH in the blood. It'll be really interesting when we get there. Organic molecules contain carbon except carbon dioxide and carbon monoxide, which are inorganic. Carbon is electroneutral. It shares electrons, it never gains or loses them, and it forms 4 covalent bonds. So this is why carbon is really essential to a lot of these organic molecules, because it's very electroneutral. You don't really need to understand why. It's a major organic component of carbohydrates, lipids, proteins, and nucleic acids. So carbon is really, I think it was the most common element in our body. Many are polymers, and a polymer is a chain of a similar unit called a monomer, which is a building block, and we'll talk about this when we get to carbohydrates. They can be synthesized, what we call with dehydration synthesis, or broken down by hydrolysis reaction. So a dehydration synthesis reaction will synthesize a polymer, and a polymer will be broken down by a hydrolysis reaction. And what that basically means, monomers will be joined together by the removal of water. So we call it a dehydration synthesis because they are being dehydrated. They are combining their OH group and a hydrogen group, and they're taking out a water because that forms an H2O. So dehydrating, taking out a water. And when that happens, they will be linked together by a covalent bond. A hydrolysis reaction is adding in a water, hydrolysis, adding in a water, and that will break it down. They'll break two monomers linked together into their 2 separate parts. So this is just showing how we're putting 2 sugars together, a glucose sugar and a fructose sugar, which are both monomers, meaning they're 1 unit. And we put them together and we form a polymer, which is a sugar called sucrose. And it's just showing how water will be taken out to form it, and we can add in water to go the opposite direction. So those are just 2 types of reactions. A dehydration synthesis reaction means you're taking water out of 2 molecules so that they can be linked together, because you're basically just taking out excess atoms. And a hydrolysis reaction means you're adding water into a molecule. And when you add more atoms into a chemical reaction, you'll get 2 separate products at the end. Carbohydrates include sugars and starches. They will contain carbon, hydrogen, and oxygen. Usually the hydrogen and oxygen are in this 2-to-1 ratio. And we have 3 different classes of carbohydrates. Really easy to see. They'll either be monosaccharides. Saccharide is a sugar. Mono means 1. So this is 1 single sugar, and a monomer is the smallest unit of a carbohydrate containing 1 single sugar, 1 monosaccharide. A disaccharide, di means 2, so these will have 2 sugars put together. And a polysaccharide, poly means many, so these will be many sugars linked together. Also known as polymers. Polymers will be made up of monomers of monosaccharides. A monosaccharide is a simple sugar containing 3 to 7 carbon atoms. The general formula will be carbons, hydrogens, and oxygens, and N will equal the number of carbon atoms. We'll have monomers of carbohydrates, meaning just 1. Important monosaccharides are what we call pentose sugars. Pentose, meaning they have 5 carbons in them. Ribose and deoxyribose are important because they make up RNA and DNA, which contain all of the genetic material in your body to create proteins. Or they'll be hexose sugars, meaning there's 6 carbons to them. So the pentose and the hexose refer to how many carbons are in this 1 monomer. I know you're probably scratching your head by now. Glucose is the important hexose sugar you could know. That's the blood sugar in your blood. So simply just know that a monosaccharide means it's 1 sugar. So these are monomers of carbohydrates, meaning these are single molecules. These are glucose, and glucose has 6 carbons in a ring. Fructose will have 5 in the ring. Galactose, or no, they'll all have 6 here. The hexoses are shown as isomers, meaning it shows just how they're arranged in space. And the pentose sugars are seen here. You don't need to know pentose and hexose. Just know that glucose is an important monosaccharide. Fructose and galactose, they're monosaccharides because they're one sugar by themselves. Deoxyribose is another monosaccharide. It's own simple sugar by itself, and ribose is a simple sugar as well. Disaccharides means we put two sugars together. Disaccharides are too large to pass through a cell membrane. Here are some important disaccharides: sucrose, maltose, and lactose. They're formed by dehydration synthesis reactions. That just means that to form a disaccharide, you put two sugars together. Easy peasy. You take two simple sugars and you put them together and they're joined together. So these are disaccharides. Sucrose is a formation of glucose and fructose together. Maltose is a fancy word for putting two glucose molecules together. And lactose is a fancy disaccharide word for putting a glucose and a galactose bonded together. Then polysaccharides are just many sugars put together. They're formed by dehydration synthesis reaction by just putting lots of one sugars together. And important polysaccharides in your body are starch. In the body, starch is the way that your body stores, not starch, this is in plants, it stores carbohydrates in plants. Glycogen is the way we store carbohydrates in our liver specifically. In you guys and in me, in animals, we store glycogen in the liver, and glycogen is just a lot of glucose molecules linked together. They are not very soluble, meaning they will not dissolve easily. So this is showing glycogen. It's a polysaccharide because it's many sugars linked together. You can see these little hexagon-like shapes here, and they're all linked together, and these are all glucose molecules that are just linked together. They get linked together because when glucose needs to be taken out of your blood, if there's too much sugar in your blood, glucose gets taken out of your blood and stored as glycogen in your liver. And that is the result of insulin production. Insulin gets sent to your blood and it takes glucose out of the blood and takes it to the liver to be stored. That's why insulin is important. And it gets kind of crazy if you're a diabetic and your cells don't respond to insulin or your cells don't make enough insulin. I'll stop going off on tangents here. Yeah.
So I know when people refer to bread, they usually say like carbohydrates. So what would that be? Would that be a I'm sorry, I don't know the word, polyesteride?
Polysaccharide. Yeah, so bread is just sugar. Bread is a carbohydrate, and carbohydrates are just sugars linked together.
Okay.
Yeah, that's why. So, but sugar again, gets a bad rap because glucose is sugar. But you'll learn that all of your cells need glucose to create ATP. So, bread, carbohydrates are not bad, but yeah, bread, specifically carbohydrates, are just links of glucose molecules together. There's different types of bread, and some breads obviously are better than others if they contain a lot of other nutrients in them. Yeah, but bread is not necessarily bad in itself because we all need glucose. Good question. You should be a nutritionist, Mia. You have all these nutrition questions. And there's probably people in here that know more about nutrition than I do. So feel free to respond to any of that. Lipids contain carbon, hydrogen, and oxygen, but less carbohydrates, and they sometimes contain phosphorus. They're insoluble in water. That means they don't dissolve. I think I misspoke. Carbohydrates, they're not very soluble. So yeah, they're not very dissolved. They can't dissolve very well easily. These are the main types of lipids. And I think one of your lecture questions, I have a matching question for what a triglyceride looks like, what a phospholipid looks like, and what a steroid looks like, and what a carbohydrate looks like. So in your lecture exam, and I'll show you an example of this, I'll give you pictures of these different types of molecules in the body. You just have to match up what the pictures look like. So I don't try to ask you too many more details other than that. So let's talk about triglycerides first. Again, these are lipids. Triglycerides are fat when solid and oils when liquid. They're composed of 3 fatty acids bonded to a glycerol molecule. Glycerol is a sugar alcohol because sugar means glucose and alcohol always will end in an OL. Again, chemistry is like a new language. Main functions to triglycerides: they will store energy, they'll help insulate the body, your fat, and they'll also help to protect your organs with a kind of a fatty layer around them. So this is what glycerides look like. They consist of glycerol, which is shown up here, which is just kind of a sugar with an extra hydroxyl group, an alcohol group, and a fatty acid tail. So this is a triglyceride, and 3 fatty acid tails. So if we look here, triglycerides will have these 3 fatty acid tails to them. So these are triglycerides. Triglycerides consist of glycerol and 3 fatty acids. So here is the triglyceride itself and the 3 fatty acid tails. So it's just kind of a combined version. It's just a simplified version of what you're seeing in this slide, and they're just simplifying it down to show this. So a glycerol molecule at the top and 3 fatty acid tails. This is a triglyceride. Tri meaning 3 fatty acid. This is a great animation kind of explaining the different fats, and you can see them a little bit shown here, but again, I'll let you guys watch this on your own, but you can download the PowerPoint. You can't watch it if you just watch it in Canvas. You actually have to download the PowerPoint to see it. Triglycerides can be saturated or unsaturated, and you've probably heard of this. Saturated fat is bad for you, unsaturated fat is good for you, and now you're going to learn why. Saturated fat is bad for you because the links of fatty acid tails are saturated, and I'll show a picture. They're all saturated with hydrogen, and they're all connected by single covalent bonds, and they create linear molecules which can pack together more closely, which form a solid at room temperature, like animal fats and butter. So this is a look at saturated fat. It's a triglyceride that has this nice kind of crink to it. And you can see how all of these fatty acid tails can be stacked really nicely on top of each other because they're all linear. They're all nice and straight. They're nice and straight because they don't have any double bonds between the carbons. So saturated fat is bad, quote unquote, bad for you. Again, I say that because I firmly believe anything in moderation is fine. But saturated fats will stack more tightly on top of each other so that at room temperature, you get a stick of butter. The same thing happens in your body. It's harder to break down saturated fat because they're so nicely packed together. Unsaturated fatty acids really simply have double bonds in them, which cause a kink in their fatty acid tail so they cannot pack together closely. So they will be liquid at room temperature. An example of these is olive oil. Trans fat is what we call modified unsaturated fatty oils that resemble the structure of saturated fats, and they're considered unhealthy. If we say something's omega-3, we say that they're heart healthy for fatty acids. So I'm going to show you what unsaturated fat looks like. See this double bond right here and see how it causes the whole fatty acid tail to like crink down. All of a sudden these fatty acid tails aren't completely linear and they can't all stack parallelly nicely on top of each other. So these kinks mean that they'll be liquid at room temperature, and that'll be easier for your body to digest as well. So olive oil is like that. Phospholipids will be modified triglycerides. You might have heard of phospholipids in the cell membrane. Phospholipids are they have a head and a tail region. The head region is polar and loves water, so we call it hydrophilic. Philic prefix means love or attraction. The tails of phospholipids are nonpolar, and they're hydrophobic. Phobic, they're afraid of water. So they're important in cell membrane structure. Remember the head region will face, the phosphate group of the head polar region will face water in the cell membrane. And the tail region, the nonpolar tail, will kind of reach up and touch each other. And this is the structure of the cell membrane, showing how the polar heads will face the exterior of the cell and the interior of the cell, that's mainly made up of water. So they'll form a lipid bilayer in the cell membrane, and the tails will face each other because they are hydrophobic. They don't like water. So they'll hide away from the water and they'll face each other. So these are phospholipids and how they will make up the cell membrane. Steroids are another type of lipid. They consist of 4 interlocking ring structures. So steroids, I'm just going to show you their structure because there might be a question on your lecture exam that says, match this structure molecule and what is its name? And steroids are easy to spot because they're just made up of 4 rings. So you can see 3 hexagons here and a pentagon. Those are just 4 rings that make up the steroid structure. And the main steroid that we'll talk about is cholesterol, and that's actually shown here. Cholesterol is the basis for all steroids formed in the body. Cholesterol is made by your liver, and it's also found in animal products like cheese, eggs, meat. Cholesterol is the starting material for synthesizing vitamin D, steroid hormones, and also your bile salts. And cholesterol is also important in your cell plasma membrane structure. So that's cholesterol. It's just a type. It's the most important steroid in your body. Icosanoids, I think you can say it a couple different ways. They're derived from a fatty acid found in cell membranes. The most important type of them are prostaglandins, and they are important in blood clotting, the control of blood pressure, inflammation, and labor contractions. The inflammatory actions are actually blocked by what we call NSAIDs, which are non-steroidal anti-inflammatory drugs. So that's what NSAID stands for, non-steroidal anti-inflammatory drugs. They're non-steroidal because they're not a steroid. They're a different type of fatty acids. So that's why they give them that name. And they're anti-inflammatory because these guys play a role in inflammation. So aspirin and ibuprofen are big ones. Proteins comprise of 20 to 30% of cell mass. They have most varied functions of any molecule. So your proteins will do everything in your body. They'll be important in structural things in your cells. They make up enzymes for chemical reactions, and they'll help the contraction of muscles. So proteins are very diverse. Polymers of amino acid monomers are held together by peptide bonds to make proteins. So proteins are made up of amino acids, and they're held together by peptide bonds. The shape and function is due to 4 structural levels of proteins. And we'll talk about those kind of structural levels in a second. Structural proteins, an example is collagen. And collagen is the single most abundant protein in the body because it helps make up bones, tendons, and ligaments. So that's collagen, a type of structural protein. Enzyme proteins. Enzymes are important for chemical reactions. They are catalysts. And then transport proteins help in moving substances. And hemoglobin is an important transport protein in your blood. And hemoglobin transports oxygen in your blood. Contractile proteins, more types of proteins. Actin and myosin are the 2 types of proteins that will latch onto each other, causing muscle contraction or shortening. Communication proteins are in the cell membrane. They can act as chemical messengers like hormones or receptors. Defensive proteins are antibodies. So you can see here how there's a wide variety of proteins in your body. But all proteins are made up of 20 types of amino acids. So if you leave with anything tonight, and I know you're probably lost already, and maybe your heads are spinning, proteins are made up of 20 types of amino acids, and they're joined together by peptide bonds. And I'm just going to keep going through that. The amino acids are all different by a different group that is on them. So this is an amino acid, and this is the R group. So every amino acid has this general structure, but the R stands for a different chemical group that gets added to it to change it. So there's 20 types of amino acids. They're all formed together by what we call peptide bonds. So we call a dipeptide is 2 amino acids bonded together. Really simply, the structural levels of proteins, there can be a primary structure, which is a linear sequence of amino acids. That just means amino acids in order. A secondary structure of amino acids is how they will interact with each other. So this is when the amino acids might coil together to form like a spring, or a beaded sheet is just a pleated sheet, like an accordion ribbon. So primary structure is just a line of amino acids. Secondary is how they're bonded or interacted with each other. Tertiary is how the alpha or beta sheets will interact with each other. And then quaternary structure of proteins is how different polypeptides will act with each other. What am I talking about here? So this is a primary level structure of amino acids. This is just the primary structure of proteins, amino acids in a straight line. Primary structure, this line of amino acids will go on to form a secondary structure where that line of amino acids will interact with each other in an alpha helix like DNA, or in a beta sheet where they'll kind of be like accordion together. The tertiary structure is how those alpha helices and beta sheets will then interact with each other. So they'll kind of be folded over. And then the quaternary structure involves 4 identical subunits formed to form a complex protein. This is a great video to watch if you're still kind of confused with this all protein structure and the 4 levels of that. Fibrous and globular proteins. I'm not going to get into too much of this. Different types of fibrous proteins are keratin, elastin, and collagen. Globular proteins are antibodies, hormones, and enzymes. Denaturization is when we have a globular protein that unfolds and loses its shape. So if a protein gets denatured, that just means it loses its shape, and they're not able to do their activity anymore. Fibrous proteins can be more stable, and their active sites can become what we call deactivated. And this can be caused by increased acidity or decreased pH. It's irreversible if changes are extreme. And an example of this, if we denature a protein or change its shape, it can't go back to what it once was. And an example of this, think about cooking an egg. You can't go back and put the egg back in its previous form. So that's what denaturing a protein means. It'll just become inactive. Enzymes are globular proteins that act as biological catalysts. So we talked a little bit about catalysts before. What they do is they just help a chemical reaction to go more smoothly and more quickly. They will not get used up in the reaction. Oh, question?
No, he's not dead.
I'm gonna go ahead and mute. Okay. These are some characteristics of enzymes. I'm not gonna ask you too many of these, so I'm just gonna go over this part a little bit. The big thing you should know about enzymes is they just lower the activation energy, which is the energy needed to initiate a chemical reaction. To do that, I'm just gonna show you, if you think about a chemical reaction occurring, and you think about this hump is the activation energy needed for the reactants to turn into their products, all the enzyme does is it lowers that activation energy required to get those reactants to form products. So they're basically bringing down this hump that the reactants need to get over to form a product. That's what enzymes do. They lower the activation energy. That's the main thing I want you to remember about enzymes. A mechanism of an enzyme, it has what we call active sites where the reactants will bind. And with the help of the enzyme, it'll form this enzyme substrate complex. And that's just the complex that is formed for the enzyme to help the reaction to occur. And then the product will be released from those active sites as the newly formed peptide bond or whatever it's trying to help create. And this just takes you through those steps there. Nucleic acids here, I'm just gonna check my time to make sure. Okay. Nucleic acids are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. These are the largest molecules in the body. The nucleic acid polymers are made up of monomers called nucleotides. And this is what a nucleotide is. And we're gonna talk about a nucleotide. The 2 major nucleic acids in the body are DNA and RNA. And DNA and RNA carry your genetic material to create proteins. No problem, Vincent. DNA and RNA carry the genetic material to create proteins in the body. DNA is all of the genetic blueprint for the synthesis of all proteins. And remember, when we're talking about proteins here, protein means your bone, your collagen, your ligaments, the antibodies in your blood. It means portions of your cell membrane. So some people, you got to remember when we're talking about proteins here, we're talking about really all aspects of your body pretty much. And your DNA holds all of the genetic blueprint for that. So when DNA gets passed down through its base pairs, if there's one mutation or kind of bad thing in that DNA, it can cause really bad problems for the person. Or if traits get passed down, the traits will be held in the DNA because those traits will create proteins that will perform a specific function or give a hair color or some sort of attribute. DNA is a double helical molecule. It's made up of nucleotides and one of 4 nitrogen bases: adenine, guanine, cytosine, and thymine. They follow a complementary base pair rules where adenine will always pair with thymine and guanine will always pair with cytosine. And these are the base pairs that'll pair together. This is the structure of DNA. It's this double helical strand. And this shows how the base pairs are bonded together through hydrogen bonds. We'll get more into how DNA is replicated later, but this is just the structure of DNA. RNA will link DNA to protein synthesis, and we'll again go through what protein synthesis is, but we're just going to look at what RNA looks like now. It will contain a ribose sugar instead of deoxyribose, and instead of the thymine base, it will have the uracil base. These are the different types of RNA that carry out the DNA orders for creating proteins, and again, we won't get into how that happens tonight. We just want to look a little bit at their structures. And this will be a great video to watch to explain the differences between DNA and RNA. ATP is kind of the last molecule we'll talk about tonight. Chemical energy will be released when glucose is broken down and is captured to make ATP. And this is the main source of energy in the body. You need ATP to power all of your chemical reactions. And the structure of ATP looks like this: an adenine group, a ribose group, and then 3 phosphate groups. So that's why it's called adenosine triphosphate. We can get an adenosine diphosphate if only 2 phosphate groups are attached, and you can see those in brackets here, or you can get adenosine monophosphate with 1 phosphate group. Adenosine triphosphate, or ATP, gives energy to power all of our chemical reactions when these bonds are broken in between the phosphate groups. So ATP, loss of a phosphate group will convert ATP to ADP, and another phosphate group will convert ADP to AMP, or monophosphate. This is the hydrolysis of ATP. So adding in water will break apart the bonds of ATP into an ADP, a phosphate group, and energy. Again, ATP is our body's energy currency because when we break the bonds between these phosphate groups, we'll release energy when we do that. So that's why ATP is important for the body. This is work driven by energy from ATP, showing in this case the sodium-potassium pump. This shows mechanical work where ATP will phosphorylate or give up a phosphate group to help contraction. And also it provides chemical work when it phosphorylates reactants, meaning it'll add in a phosphate group to a reactant. And that's the end of this whole chapter, I guess, part 2.